Hello students, welcome you all to a new video of Sunanda's tutorial. In this video we will discuss about the enzyme inhibition. So what is enzyme inhibition? So this is a phenomenon where a molecule called as inhibitor binds to an enzyme and stops it from working or slows it down. So enzymes are generally helpers to and help to speed up the chemical reaction inside our cells, but some molecules which are called as inhibitors stop them or stop enzymes to do their work. Inhibitors can stop enzyme in two main ways: in reversible manner and irreversible manner. Irreversible inhibitors, those inhibitors which can bind very tightly to enzyme, often making permanent chemical bonds with the enzyme and finally destroy the enzyme forever. So those are called as irreversible inhibitors. Reversible inhibitors, they bind loosely and can detach or pop off later on. They only stop enzymes temporarily, enzymatic action temporarily. Okay. So there are many reversible type of inhibitors, and the first one is competitive inhibitor or competitive inhibition, which is a process where molecule which are structurally similar with the substrate binds to enzyme's active site, finally blocking the actual substrate from attaching. So this kind of inhibitor is completely looks like the substrate, original substrate of that enzyme. So they attach on the active site of an enzyme. Because this binding is reversible, the inhibition can be overcome by supplying or flooding the system with high concentration of original substrate. So this competitive inhibition can be overcome or can be reversed by adding more substrate on that system. Mechanism: the inhibitor and the substrate compete for the exact same active site on that enzyme. Only one can bind at a time. Okay, so this is the case of competitive inhibition. Next, non-competitive inhibition. Non-competitive inhibition is a type of enzyme regulation where inhibitor binds to the site other than the active site, which is called as allosteric site. We will discuss allosteric effect later on. So here, unlike competitive inhibition, in case of non-competitive inhibition, the inhibitor attach to a site other than the actual active site of an enzyme. This binding changes the enzyme's shape, preventing it from converting substrate into product. So this inhibitor and enzyme attachment or binding finally leads to the change in enzyme's shape and which prevent the enzyme to convert the substrates into product, which ultimately decreases the reaction rate of that system. Okay. Next one is non-competitive inhibition. It is a type of inhibition or enzyme regulation where inhibitor binds to preformed enzyme substrate complex. So at first enzyme binds with the substrate, then it forms enzyme substrate complex, then finally enzyme plus product. So here in this case of non-competitive inhibition, inhibitor binds to the preformed enzyme substrate complex and prevents the complex from converting into the product and lower both maximum reaction velocity and apparent Michaelis constant. So finally they lowers down the reaction rate of that system. So this is the case of non-competitive inhibition. Now we will discuss some examples. So at first, the example of competitive inhibition. Malonate is the competitive inhibitor of an enzyme succinate dehydrogenase. So succinate is the main substrate of an enzyme succinate dehydrogenase, and malonate is the competitive inhibitor of succinate. for the enzyme succinate dehydrogenase. Next, example of non-competitive inhibition. So cyanide acts as lethal non-competitive inhibitor of cytochrome oxidase. Cytochrome oxidase is an essential enzyme in mitochondrial electron transport chain. So after binding with the cytochrome oxidase, it changes the enzyme's shape, preventing the cells from using oxygen to produce adenosine triphosphate. So the example of non-competitive inhibitor is cyanide. Cyanide for cytochrome oxidase in ETC or electron transport chain. So now the example of non-competitive inhibition. So phenylalanine binds to intestinal alkaline phosphatase. Phenylalanine only binds to the enzyme after the substrate has already bound. It essentially frees that enzyme, frees that enzyme and substrate complex and prevent the formation of the final product. So the example is phenylalanine is the non-competitive inhibitor of an enzyme intestinal alkaline phosphatase. Okay, next, what is allosteric effect? So allosteric effect is the process by which a molecule binds to protein at a location other than primary active site, or it is not protein, it is enzyme, causing the structural shape change that alters the protein's activity. This mechanism is fundamental to in regulating enzymes and proteins inside our body. What is allosteric site? It is a specific other binding site on protein or enzyme where regulator molecule attaches. Okay. What is orthosteric site? It is the main active site. What is conformational change? It is the physical twisting or shifting of protein's 3D shape, okay, which either turns the protein's function on or off. So what is allosteric regulation? Allosteric, either it will show allosteric activation or allosteric inhibition. Sometimes the allosteric molecule shows positive effect, sometimes it shows negative effect. Allosteric activator, which is the positive one, binds to the protein or enzyme, changing the active site to better fit the substrate, increasing the overall protein or enzymatic activity. But in the other case, negative modulation or allosteric inhibition, where allosteric inhibitor binds to the protein or enzyme, altering the active site so that substrate can no longer bind efficiently, so ultimately reducing or stopping the enzymatic or protein function. Another thing is their cooperativity. It is a special type of allosteric effect where the binding of substrate to an subunit of multi-unit protein like hemoglobin changes the remaining subunits, making it significantly easier or sometimes harder for subsequent substrate attachment. So these are the cases of allosteric regulation. Sometimes they affect positively, sometimes they affect negatively, sometimes many subunits act cooperatively to do one function. Okay, so these are some cases of enzyme inhibition. Another term, remember another term, which is feedback inhibition. What is feedback inhibition? Where the product of a reaction blocks the reactants to react further. When the concentration of product increases, the reactants ultimately stops to react and finally the reaction stops. So that is called as feedback inhibition. So these are different types of enzymatic inhibition. I hope this video will help you further in future, and if you liked it, please hit the like button, share with your other friends, and please subscribe to my channel Sunanda's Tutorial. Thank you.